So there's a very famous lake at the Hangzhou called the West Lake, dated back to thousands of years ago.
Hi.
So that's why our university called West Lake University, because it's just located in Hangzhou and near West Lake.
Interesting.
Yeah.
It's in the west of the city.
It seems so cool.
So later, our chair, Mr. Xiaobing, so she will be hosting this event. And after your presentation and Jushen's presentation, we have a short discussion, maybe collect some questions from the online audience. Then after that, if we have time, we can close the broadcasting and we have a private discussion of potential collaboration.
Yeah, of course, that would be nice. And that's why Mike Pietzner is there. So he's done a lot of the work that I present. So he'll be very bored. And then I think Julia Sanchez is also joining, who's done the prediction work that I present at the end. And I don't know whether Mina was invited or not. But yeah, no, we look forward to the smaller group. And before that, very happy to do the questions.
Great.
Yeah. Great. I think it's a time. So maybe is the broadcast over? Is going on now?
Yeah. Bing, are you there?
Yes, I am
Bing will
here.
come in shortly. Yeah. Okay.
Yes, I'm here. Yes, I'm right here. Okay. Thank you, everybody. Welcome to the 8th webinar, Westlake Proteomics series. We're very lucky here to have this 8th Westlake Proteomics series co-hosted by Westlake University, Westlake Laboratory, Guomics, IMARC, and the HuB and Westlake Omics. We would like to also pay a special tribute to Chinese Ministry of Science and Technology Belt and Road Initiative, innovative international talent program. We're honored to have invited 6 distinguished speakers today with us. We are very honored to invite our first speaker, Dr. Claudia Langenberg, to give the first talk on translational insights from large-scale human proteomic studies. Welcome.
Thank you very much. Thanks so much for the kind invitation to speak. And we're very excited about our potential collaborations. And I'm delighted that some members of our team are also joining. So yeah, thank you. So before I start, so we actually work on various omics layers that kind of go or move from genome to phenome in this kind of schematic flow of biological information. But for this talk, really specifically, I will take a proteocentric view, so only talk about proteomics. And I will take you through why we think proteogenomics can give insights into identifying causal genes and mechanisms, go as far as identifying shared disease causes by anchoring our investigations in the genome, and then end with some of the studies, non-genetic studies on disease prediction. And we have done a lot of work on platform, synergy and comparison, but that's not going to be the center of the talk today. So I'm, as Jusheng knows, and we've worked together in Cambridge, I'm an epidemiologist, and for most part, epidemiologists and specifically genetic epidemiologists focus and study and like large populations. And of course, if you scale up, this influences the choice of the matrix that you use, i.e. blood is very easily accessible. And it also affects the technologies that we can choose, because it has to be fast and it has to be scalable to the numbers that we need for the power or to achieve the power in the studies that we need. So of course, compared to somebody who does smaller scale specific investigations of one particular or set of particular proteins, there's a difference there in terms of the content and the specificity of what you can measure. For many of our studies, and I like to say this always at the beginning, is our aim really is to be broad as possible in terms of the coverage on the molecular scale, so coverage and scale on the molecular side, and also be, and this is what you see here in terms of depicted by the phenome, be as broad as possible in the integration of that with phenomic data or disease data or genetic studies across a whole range of different diseases across different specialties. Yeah, so that's just the outline. If you're interested in metabolomic studies, we've published several of those, and there are also webinars available. So sorry, I'm not covering this today. There we go. Sorry. Sometimes it hangs a little bit. So I think the people here interested today, maybe I don't have to convince you, but the question is why proteins? So of course, proteins have very important roles in very diverse biological processes. We know they're dysregulated in various disease states, and most important class of drug targets. Now, I've already said we focus on blood and therefore look at the circulating plasma proteome. And here what you see on the left is the very huge dynamic range where concentrations span almost 10 orders of magnitude from the very abundant end of albumin down to small signaling proteins such as interleukins. So what this means is at the moment, actually, ideally, we want to capture, I said, be as broad as possible, but currently no single technique is able to provide reliable measurements for all of the proteins that could be detected in plasma or are circulating in plasma. And before I start, maybe just a little reminder of what we actually measure. And if you consider this here, the vast majority, the vast majority of proteins that we actually do detect that are circulating are not actively secreted to blood, so such as coagulation factors or cytokines, but they're products of cell leakage and turnover or soluble fragments of membrane proteins. And that's something that's important to keep in mind because the origin of the proteins that we detect in blood is not necessarily or always as obvious. It's just important to keep that in mind. Now, this is a nice review that Julia, was the last year PhD student in the team, wrote. It's really to remind us, how do we move from protein to proteome or omics, kind of this kind of broad capture, these broad capture technologies that enable us to assess not just hundreds, but actually thousands of protein targets. So how is that achievable? And on the left, you see mass spectrometry. And I should say, actually, from the start, as I've said, I'm a clinical epidemiologist. We're users of this technology in large-scale studies and clearly see the opportunities, but we're not protein specialists. So we collaborate with people, and here for mass spectrometry, which is clearly still the gold standard, we've learned, we collaborate with Marcus Reiser, who used to be at the Crick, first in Cambridge, then the Crick, and is now leading the biochemistry at the Berlin Institute of Health. So we collaborate, where he clearly developed higher throughput methods for mass spectrometry. But it is clear from the studies that we've done so far, and we're kind of currently scaling this up, that it lacks sensitivity for the less abundant end, which includes many of the proteins we're really interested in because they're highly biologically active, such as hormones. Now, Olink, proximity extension assay, or SomaLogic's aptamer microarray, they get around this by kind of designing these tailored affinity reagents, and then they measure them at different dilution spins, and hence they're able to detect both the more abundant to the less abundant end of the proteome and capture many more proteins. So the technologies that we've used is now the slightly older version of SomaLogic, capturing or identifying or targeting 5,000 proteins. And Olink used to have these kind of bespoke small panels and have now also moved on to larger assays that capture altogether now 3,000 protein targets. One of the things that is maybe also important to remember is that the antibody-based technique, or kind of Olinks is similar to an antibody-based technique, is quite different or qualitatively different from what SomaLogic is doing, who's using these short oligonucleotides or aptamers. And the specific point about this is that these kind of are affine or have affinity to the 3D conformation of the target protein. And this is something that's quite specific. So it's something about the shape of the protein, not just simply the concentration necessarily in plasma that's reflected. So again, it's just something to keep in mind that I will come back to. So here's how we started out, and it's taken a little bit of time to kind of get to grips with some of the differences and similarities and synergies between these different platforms. But if you just consider this here, just on the right is the overlap of what we've originally measured in the Fenland study. Fenland is a population-based study near Cambridge, or in Cambridgeshire, And the kind of beauty of this study for proteomic researchers, these people are relatively young, so born between 50 and 75 at baseline when we stored the samples. And these are the samples that we used for the proteomic measurement. And people were relatively healthy, treatment naive, and hence, because we know the existing health states of health affect the proteome, that's an advantage if you're interested in genetic discovery for this, the levels not to be as affected by existing diseases. And the other opportunity is for a study of this size relatively large. So we measured all over 12,000 individuals using this SomaScan version 4 that you can see here. And these overlap to a very large degree with the 12 panels that we measured in a small subset of 500 individuals, or these were the ones 485 successfully measured. And then again, only up to now, a small experiment with Marcus's SWOT-MS, and that's capturing 340 proteins, of which only some of them overlap. But that's currently scaled up to all Finland participants. And we've also done a range of other new O-link experiments, which I'm going to briefly present at the end. So this is the results. So Mike and Ellie led this study, and this is the result really of several years of work from first kind of getting the agreement set up, sending the samples, and receiving the data. But what it really shows is the beauty that if you scale up, how you increase your ability to identify cis as well as trans-PKTL. So I'll just walk you through this very quickly. So here on the x-axis, you see the position of the genetic variant. On the y, you see the position of the protein encoding gene. And when these two converge, so when the variant that you identify is within or in close proximity of the protein encoding gene, this is the cis-related variation here in pink. And at each of these dots really is just a specific genetic position or variant in which you then kind of test differences in the intensity that you observe for the protein target. So I can't go through this in much detail. I think it's a beautiful summary of a lot of work. But the thing I really like that they've depicted here at the top is what you can see is these blue lines that go up are these hugely pleiotropic regions. So it's a specific region associated with hundreds, if not thousands, of these aptamers. And the thing I like to point out is just how this can be qualitatively different. So here's the number of associated protein targets at this specific region. So you see this here. This is actually, I think, the axis curtailed. So complement factor H here, or the MHC region. But there are some for where, and the size of this lollipop really shows how many independent genetic variants drive these hundreds or thousands of associations. And for some of these, it is a lot of independent variants, such as here, that drive these many associations. But for other examples, actually, it is only very few. And that's here are some of those examples. And that's a qualitative difference in terms of the pleiotropic signal that you observe. So it's important to notice. And then this is kind of just in some ways a listing, and it's now out of date as we and others have done larger scale studies, but we identified thousands of genomic regions that are associated with these protein targets. And many, or at the time, many of those had not previously been reported. So I want to come back to the point, why bother? Why are we excited about this? Of course, the discovery is always exciting in itself, and the biology that you can discover, but there are also important uses and applications of these findings. And I want to just talk you through a few of those. So the first I want to illustrate by here on the left, you see the kind of evolution of genome-wide association studies only until 2017. But you see, the more participants you include in the study, the greater the power, the more association signals for each of these genetic studies for a whole variety of traits you identify. And as you can see here on the right, this is kind of as of the beginning of last year, there were over 120,000 entries in the GWAS catalog, i.e. variants identified at genome-wide significance with a whole variety of diseases and traits. But the point, and I really like this kind of little illustration that Mike did, where really for the vast majority of these, we do not know the underlying causal gene or variant or mechanism, because many of these are intergenic. We have really, for the vast majority, no clue really what's going on. So, and this is one of course, one of the big stumbling blocks if you're interested in kind of translating these discoveries in experimental follow-up. And so the beauty of the pQTLs or the cis pQTLs is we're looking at variation within the protein encoding gene affecting the gene product, and hence the flow of this information is in this direction. And if we identify that this specific variant is also identified with a disease, it gives us some clue that this may be the underlying flow of information from the variant to the protein to the disease. And we can now use this systematically by looking at all of the cis pQTLs, i.e. in the protein coding gene that we've identified, and do they appear in the GWAS catalog to be associated with different diseases? And that's exactly what this says here. So there were 588 cis regions that showed associations or overlapped either the same variant or variant in very highly correlated, associated with over 500 GWAS catalog traits. And over and above what we can learn already from expression QTLs, 25% in 25% of these cases or these connections, the cis pQTL prioritized a different gene to the one that was highlighted in the GWAS catalog or was the nearest gene. And that's a pretty big number, given that this is, as I said, just one of the uses of these results, and that this is such a big limitations of translating findings. So that was very encouraging. And on top of the 24% or 25%, there was another 14% where a long list of candidate genes in a, let's say, gene dense region could be refined to a shorter list of candidate genes. So an incredibly useful approach over and above what we can find from cis eQTLs. Now, it's I should say this is in some ways a relatively quick and dirty approach, because it's looking at the variants which are overlapping, which to some degree could still be coincidental. So this is what I mean by coincidental, where actually the genetic signal here for my one trait, i.e. the protein, and here the disease, actually are not driven by the same causal variant. So this can be tested if you look at the whole association pattern in the region by testing, is it actually driven by a same underlying causal variant? Is the signal shared between these two traits? And then you get a probability of this being a shared genetic signal. So it's going further than simply taking lead variants or proxies and overlapping between the molecular as well as the disease trait. So, and then here, and again, I come back to the advantage that the protein data give you. Here we see the signal is overlapping. It's a shared genetic signal between these two traits. So let's say it's higher posterior probability of these two really being driven by the same genetic signal. Now, if we look at two complex traits, let's say insulin and adiposity, the direction of travel is not always clear, even if this evidence is there. However, if we look at the protein encoding gene to the disease, that flow, biological flow that I started with, is given, and hence it gives us more confidence of that this is the direction of travel. So this comes back to what I said very early on, that we don't actually often know where the protein detected in blood actually originates from. And so this is another important use of the tissue expression data, which is often shared. So the GTEx consortium, you'll all be aware of, that shares the data across 49 different tissues. And there's also the eQTL-Gen consortium for whole blood, which is more powered, but only, of course, whole blood. So we systematically integrated this. And on the left here, you see what I've said earlier, that this is the nature of the somologic proteins that protein targets that we had measured. And you see that only 11 or 12% of these are actually actively secreted to blood. So the vast majority are not secreted. So we can use this tissue expression data to try and infer the tissue of origin of these plasma proteins that we detect. And that's what we then did. And in fact, so this can be done using the same technique, so that I've outlined earlier, using co-localization, in this case, not using the disease data, but the eQTL data. So just systematically integrating the genetic signal we see for the eQTL with the pQTL data. And if you do this, you see that for 50% of cis pQTLs, there's strong evidence of co-localization with a cis eQTL and or a cis splice variant. And for 145 proteins, there was evidence for a tissue-specific effect. So that comes back to what I said earlier. If it is unclear or not known where the protein originates from that we detect in blood, if we see tissue-specific evidence in this co-localization analysis, that means we can infer directly whether it may come, let's say, from liver or other tissues. So that can be incredibly helpful to identify the origin. Now, so I want to move on to kind of integration of the phenome, and in some ways it's exactly the same as I've outlined before. So we take the protein encoding gene, and we can now integrate the data for genetic studies of a whole variety of diseases. And it is one of the, again, one of the huge opportunities that the genetic data affords us, is that because so much of the genetic data for a whole variety of phenotypes and diseases is publicly available, it gives us this opportunity to do a genetically anchored study to try and infer potential causal mechanisms by basically integrating this data that comes from a whole variety of sources. So very grateful for all the people who share this data and enable these investigations. So so one of the first examples I want to show you here is not the first that we did, but it's just a nice example for where it's kind of a specific, I shouldn't say disease, but condition in this case, the prognosis for an infectious disease. So this is using publicly available data from the COVID-19 host genetics initiative. And they have different outcomes that they analyzed, ranging from infection to hospitalization, severe COVID. But what we identified using this open access data, looking at these different COVID-related outcomes, is that F5, so the ETS-related transcription factor F5, was the most robust candidate protein that co-localizes on chromosome 11 with particularly severe outcomes of COVID infection, with an odds ratio of almost 5. So for a common variant, this is a relatively sizable effect. And more so, it co-localized with a cis EQTR specifically in the lung using the GTEx data, which is what you see here. So these are these kind of local regional plots where you can kind of see what I've described earlier, this kind of, you see the association pattern with the lead variant here for the transcript in lung, and then overlaid the protein signal, and then overlaid these the associations in the genetic data from the COVID-19 HGI. So, and what you can visually see is what I described verbally, is this kind of shared genetic signal where you can really see already from the shape of the association without even formally testing, is that this is driven by the same variant. And in fact, if you then integrate this together and do what's called a multi-trait co-localization, it prioritized the lead variant that we had seen for the protein as the likely causa variant. Now, at the BIH, the group by Christian Conrad and actually Søren Lucassen and Lawrence Chua is a PhD student in Christian's group, they work on single-cell RNA-seq data. And of course, during COVID, that was also the case for various sites of the respiratory system where they had collected data in addition to publicly available data. And so we collaborated, or we approached them, Mike approached them, to kind of try and use their and the publicly available single-cell RNA-seq data to look at the expression at different sites of the respiratory system. So this is olfactory mucosa, nasopharynx, and lung. And what they demonstrated was that L5 is highly expressed in epithelial cells that are also the target of SARS-CoV. And I'm not sure this is so obvious to if you're not used to these plots, but what it also at least suggested is that L5 expression in cystenticular cells correlated with TMPRSS2 expression. So you can kind of make out from, if you compare the F5 expression with the ACE2 here down here. So I just want to give you this as an example where, using a totally agnostic approach, but using a genomic approach to anchor data from a whole variety of sources, you can come to kind of a very robust association that you can take forward as far as kind of in terms of tissue-specific cell expression for the candidate gene, which I think is is a nice example of the various uses of these kind of data and studies. So, but kind of importantly, I've shown you this now for one disease or different outcomes related to the same disease. But of course, in theory, this is kind of absolutely expandable. And this is what we have done here. So kind of really integrated genetic protein signals with genetic data on a whole variety of diseases and continuous traits that go across, as you can see here, most clinical specialties. So really any data that was available at scale and genome-wide. And you can then pull this together based on the posterior probability that you achieve of these being a shared signal. So you have then a map, proteogenomic map, that goes from the gene to the protein signal to any disease that co-localizes with or shares the same genetic signal that you identify around or within the protein encoding genes. And what this is, sorry, I think this is, no, sorry, here is just really the graphical display of this map. But I just take you through just one example or two to show you the possibility that you have here. So here you see diseases, and in the middle, kind of on the left, you see diseases, and here continuous traits that are ordered by the number of proteins with a shared genetic signal. So you see here, coronary artery disease shows the largest number of co-localizing protein targets. And then here on the right, you kind of see the opposite, the proteins which are ordered by the number of phenotype associations that they have. And so these are extremely pleiotropic signals, several of which you see at the top are those that you will recognize as being known because they've appeared in a whole variety of GWAS already. And I just want to take you to one of the ones that is kind of one, not the, but one of the most extreme examples. So here, this is now coming to this graphical display of the kind of more than 1,800 connections that were drawn for the diseases as well as continuous traits. And here on the right is kind of just zooming in into this one example, which is this kind of fibrillin 3 centered subnetwork, where we saw this convergence of kind of 37 diverse, and this is kind of a loose term that I use for this, soft tissue disorders as well as anthropometric traits through this fibrillin 3 protein, which is an extracellular matrix glycoprotein, which is encoded by FMP1. And here I show you this just in a little bit more detail again with this regional plot that zooms into here the protein encoding gene. And this is where you can see here, so the genetic signal for the protein co-localizing with expression in subcutaneous adipose tissue. And then here, just some examples, because not all of them fitted on the plot, but these various, as I said, loosely kind of diseases and traits related to elastic fiber morphology or abnormality in that. So in a sense, associated with hypermobility or abnormal elasticity of the tissue, so that then leads to hernias, has greater lung function, and a greater risk of varicose veins, taller height, but actually the opposite for diseases that are related to kind of constrictive tissue disorders, such as carpal tunnel syndrome, and I think actually as well even surgery for independent set surgery for carpal tunnel syndrome. So a really nice example in my view, and how, again, an agnostic approach can pull together, you know, etiologically seemingly related diseases and anchors this in a specific gene and potential mechanism that otherwise would have been very hard to identify. And then on top of that, not all of the features of the FMP1 knockout mouse, but several, such as hernias, were also in line with what we had seen using the human genetic data. So I'm going to end this part soon. I'm just going to quickly check the time because I'm running out of time. I just wanted to highlight for those of you who are interested. So Mike has done extensive work on the comparability of the Olink and SomaLogic platforms. So if you're interested, please read up on this. And then Mina Popoul is a student in our group, and she's currently doing a whole variety of work, pulling together data that exists in a consortium called Scallop that's focusing on these Olink panels, which you see here listed. So a large amount of the data she's pulled together, QC'd, meta-analyzed, are samples that have not previously been published. And we're doing this in an effort to kind of anticipate the release of the Olink data in UK Biobank, to have the largest platform-specific meta-analysis for any of these proteins. And in parallel, we're doing a new SomaLogic large-scale meta-analysis. So ultimately, for any given protein, be able to pull all of the data together across platforms. And of course, for some of these, this will be feasible. For others, it may be slightly more challenging, as Mike's work has already shown. And then again, I really, for this, only want to highlight the preprint that Mina has already published, which anticipates the release and the pharma consortium results on the Olink Explore and Expand, these 3,000 proteins that can be measured, where she's really gone through the whole motion of not just the genetic discovery, but integrating this through statistical co-localization with a whole variety of disease endpoints, and then using this to compare where we see signals for the common cis pQTLs, whether or not this converges with what we see for the rare variant gene burden for those same diseases for these genes. And then on top, as I said earlier, to also use this to try and identify causal candidate genes for existing GWAS loci. And in fact, here for Olink, the proportion of re-annotated causal genes is even higher than what we had originally observed with SomaLogic, with the 24% that I outlined. So I'm going to change tack and just finish in the last few minutes with some of the work that we have done, which is a different use of this technology, which is disease prediction. So this is an early study that was really driven by SomaLogic, who we kind of did this in collaboration in the Finland study, as I said. And there's just a few things I want to highlight for these prediction studies. So in this endeavor is mainly cardiometabolic traits and diseases, cardiovascular disease, diabetes, and kind of the development of kind of commercial insights, that kind of protein signatures that can help to predict these diseases. And what the study basically shows is that this is possible. Proteins only are pretty good at predicting these various cardiometabolic traits. But there are a few points to make with this, and there are now other studies as well for specific diseases. So far, this is really limited to selected diseases that people have studied in a given setting. And for many of the diseases that people have predicted, there are those that are actually relatively easily and cheaply predicted, but that is not the point. The opposite is the point. The point is what you want to predict are the entities or the diseases or presentations that are otherwise extremely hard to identify or overlooked. So that's the first point. The second one is that, of course, if you're a proteomic company, it makes sense to develop extremely expansive and expensive protein signatures. But again, that's not the point. In a healthcare setting, what you want is an extremely sparse model, i.e. the smallest number of informative proteins to predict a given entity. So again, that's another concept I think that's largely been ignored. And then finally, this is generally disease specific. Nobody looks across different diseases and whether you can identify proteins that may have an ability to predict not one, but several diseases or linked diseases. And again, if you think of other predictors aren't necessarily causal, but consequences of diseases. And many predictors that we know are actually already shared. So this is also the case for proteins. So I can't go through all of the work. I just want to give you one example and then I end. So this comes back to the first point of predicting an entity that is otherwise overlooked. And this is an interest of Julia's. As I said, finally, is a PhD student, and it's isolated post-challenge hyperglycemia, which is actually also very common in Asian populations. So it's glucose levels that are elevated, not fasting, but 2 hours after you give people an oral glucose tolerance test. So in a sense, you give people a can of Coke, and then they have this inability of bringing these levels down after they have had this challenge. And It's strongly predictive of cardiometabolic diseases, more so than a fasting glucose is. It also increases much more with age, unlike fasting glucose, which is relatively stable over the life course. Is rarely measured because you have to do this challenge test, and that takes time and it's annoying. So it's not a first point. It's not the test that people do when people first present. It is, however, extremely common. 7.5% of adults globally. So it's a huge number given how large a risk factor it is for the development of diabetes, as well as other cardiometabolic complications. So isolated post-challenge hypoglycemia is an entity that if you measure the fasting plasma glucose or HbA1c, and they're both in the normal range, and is simply only the 2-hour value that is elevated. So the question is really, it's a simple question. Can we identify a proteomic signature using a fasted sample, or actually a convenient sample, to design a simple test that predicts this entity? And we were lucky enough in the Finland study that on everybody who participated, as Zhisheng will know, there was a 75-gram OGTT, or glucose tolerance test, and we were able to identify people who had impaired glucose tolerance or isolated impaired glucose tolerance. And then Julia used a machine learning approach, a lasso regression, where she divided the Finland study into these 3 sets, where half for the feature selection, then a quarter for parameter optimization, and a quarter for where she evaluated the model performance, i.e. in a set that hadn't been seen by the part that selected the features originally. So I'm just going to skip to the results. And if you just simply focus on the right, because that's the isolated IGT, it's another point that's important. If you want to look at prediction, it's very important to consider whether for this, for the clinical entity you're interested in, there is already a gold standard model. And in this case, of course, what people would do is to measure fasting plasma glucose and HbA1c to identify people. So, and this is what we set out to test ourselves against the toughest possible benchmark. So in yellow, you see here the AUC that is achieved by using a non-invasive or kind of basic patient information risk factor like age, sex, BMI, and so on. And then on top, you do fasting plasma glucose and HbA1c. So it's a serious 0.75. But actually, 73 proteins alone already vastly outperform here in green, even this kind of benchmark or best clinical model. And then coming back to the question of a sparse model, Julia then asked, what can be achieved if we only add the 3 most informative proteins? And that's the blue line, and that's still an AUC of 0.82, substantially larger, substantially and significantly greater than what you achieve with the kind of gold benchmark. And this leads to a net reclassification index of 16.5%. So that's relatively sizable, as the ones of you who do prediction studies will appreciate. So kind of a more appropriate classification of people in 16.5%. And it's largely due to the number of cases, i.e. people at higher risk being reclassified rather than the number of controls. So yeah, clearly this has worked, and this has now inspired us to look at prediction, addressing the points I said earlier across a whole range of diverse diseases where we can see does this work more broadly, and also specifically can we identify proteins that may be predictive of not one but several otherwise hard to predict diseases. So with this, I would like to end. I want to highlight so that we share our results in this kind of omic science web server. And we're very grateful to our colleagues at the Helmholtz Center, Gabi Kastmüller, Maria Wörheide, who've kind of really helped. This is across different metabolomic proteomic studies. So if you're interested, you can go, you can explore targets, you can look at the network, you can download all the data for your use if you're a scientist or in an academic setting. So in summary, sorry, this was a little bit of a rush through, but I hope that I've shown you the potential of omic technologies for translation and how it can really help to better understand disease mechanisms and identify disease specific as well as disease shared disease causes, and that there are useful applications for disease prediction. And finally, much of that would have not been possible through close and really good collaboration with industry and specifically various proteomic companies and proteomic academic groups. So I hope I highlighted the people who've done all the hard work throughout the talk with their photos, but there are many people from my various institutions that I have to thank here. And last but not least, Nick Wareham, who was the co-lead of the group at the MRC Epidemiology unit for a long time. And I've only recently moved my institution from Cambridge, where I was a long time, and then to Berlin, now to Queen Mary University of London. So thank you to all my colleagues. This work was all done mainly when I was at the University of Cambridge. And of course, thank all of you for your attention. And if you're interested and you like what you saw, currently at QMUL and at BIH, we're hiring, so particularly statistical geneticists. So if you're interested, please email me. And thank you for your attention.
Thank you, Dr. Allegenberg, for your exciting talk. We can see how proteomics can be a powerful tool in helping find treatments or cures or predict challenging diseases, such as highly prevalent diseases, like you mentioned, carpal tunnel syndrome and diabetes. And also thank you for building powerful tools such as omics science to share with the omics scientific community. We'll probably have a lot of questions from audiences and panelists or speakers today, but we'll have the second talk and then we'll move on to the panel discussion to have these beautiful sharings. So coming up next, we're very honored to have invited Dr. Zhengju Sheng, who will give a talk, and he's one of the PIs at Westlake Omics. Welcome, Dr. Zhengju Sheng. Welcome here.
Thanks, Xiaobing. So I thought Claudia will have a Q&A first, but anyway, I will have a relatively short talk to echo Claudia's work in the European population, and we have more time for the panel discussion and the Q&A. Let me share my screen. So yeah, first of all, I will really thank Claudia to accept our invitation to give this talk. It's really inspiring because all my group has followed Claudia, your work in the past few years. It's really a lot of interesting work integrating genetics, proteomics, and to explain the normal biomarkers for human disease. So our work is mainly on focused on Chinese population, which we found that is really have anyone to integrate the blood proteomics with human cohort data in Chinese population. When I established my own group four years ago at the Westlake University. So I think I will skip this background introduction about the importance of blood proteomics for human health, like drug discovery, predict biomarkers discovery, etc. And this slide also has been introduced by Claudia, because this is from Claudia's group review paper. So far, blood proteomics research is really really hot topic, and there are several ways to measure blood proteome in terms of high throughput, which high throughput actually is essentially a large scale cohort study for the blood proteomics measurement. So there are two methods, basically a mass spec based and affinity based. So now in the European populations, which Claudia mentioned, the tens of thousands of people, they mainly use the affinity based method to measure this blood proteome, which is really high efficient, I would say. But there's really any large scale mass spec based proteomics research. And of course, there are pros and cons across different methodology, like this epidemiological or immunoassay-based, affinity-based method. They could simultaneously measure a lot of proteins, like Olink or SomaScan. And the mass spec method, somehow the limitation is that we can only measure around 500 proteins, but it has another strength of sustainability, which we can reuse this mass spec data by using novel evidence in future, which means that the current 500 protein could expand to over 1,000 in next few years if the evidence improved. So Then we collaborate with Tiannan and and a colleague at the Shenyang State University, working on this particular cohort study for the Guangzhou Nutrition Health Study. So this is a study in Guangzhou, southern China, involving over 4,000 participants. So over time, I think it's established in 2008, and follow these participants are followed every 3 years with blood sample collected every 3 years over the past 10 years. We collaborated with Tiannan and Professor Yu Chen at Guangzhou to initiate this GNH or Guangzhou Nutrition Study Proteomics study. Basically, what we want is using blood sample collected over 3 time points over the past 9 years, and to measure the blood proteome using the serial samples. So overall, we have now measured I think around 7,500 serial blood proteome data among these 3,700, around 800 participants over 9 years follow-up. So we have another validation cohort. So we divide our cohort into discovery and internal application because we measure this blood proteome in different phases and including a pilot study, which I'll mention later. So to test the feasibility of this kind of large-scale mass spec-based proteomics research in our cohort, we initiate an investigating control study. So I will introduce later. So the QC of our own, the whole kind of DIA mass spec proteomics are done mainly led by Tiannan's group. PhD student Xue Cai did all this stuff, and their methodology paper, I think, is recently published in Nature Protocol. So we have followed a very rigorous QC procedure, including the biological replicates and QC samples. I'm not an expert in the mass spec proteomics, but Cai is in the panelist. So if the audience has any question with regard to the QC of this DIA mass spec-based proteomics, I think Xue can help explain. But anyway, the pilot study dates back to 3 years ago. So we, based on our Guangzhou Nutrition study, we designed a very small-scale nested case control study for type 2 diabetes. So we selected 200 and 800 incident diabetes case during around 7 years follow-up. So we picked around 200 samples. Those case and controls repeatedly measured their blood samples twice over the around 5 years follow-up. So and then we have another small-scale validation study. So this pilot actually worked very well. So we kind of depict the blood proteome trajectory, the change over time from baseline to first follow-up of 3 years and another 3 years second follow-up. And we successfully identified 7 co-protein, stereo protein, which is predictive of type 2 incident type 2 diabetes. And actually includes 3 well-known diabetes-related protein, including the sex hormone binding protein at the top, contributor or predictor for incident diabetes, and the ApoF and the CFH well-known. The other 4 are relatively new, which have never been reported to connected to incident diabetes. So based on this 7 stereo protein, we developed a protein risk score, and using the risk score to see whether the overall score are connected to diabetes incidence, and we found whether the both the baseline protein risk score associated with future incident diabetes, but as well when we use the repeated measuring data with the linear mixed model, we also confirm that the change of this blood protein risk score also connected with incident type 2 diabetes. So that's kind of a very initial pilot study that back 7 years ago. So our next interesting point is have something in common with Claudia's group. So we are interested in linking the genetic and the proteome to see whether there's we can identify some Chinese people specific, maybe drug target or intervention target for human disease, because the integration of host genome with the blood proteome have a greater potential for this newly biomarker discovery or drug target discovery, or we can find a novel connection with blood protein with human disease. So we start this based on this collected blood proteome data. We run this PQTL study. So it's led by a PhD student from in my group, which has done a lot of excellent work. So Guangzhou Nutrition Health Study is our discovery cohort, with around 2,400 people with both genetic data and the blood proteome data available. Then we use another 500 people from Westlake Precision Birth cohort also is a cohort established at the Westlake as a replication. So we did those PQT protein wide genome wide associated study, and also did some chance estimate comparison, and then do causal inference for human common disease. So overall, our Chinese speakers, this probably was the first Chinese PQTL study for blood proteomics data. So it involved around 3,000 people, and using the DI-based protein as I introduced before. So this is Manhattan for overall results from PQTL study. So as I already introduced, the protein measured by the mass spec is somehow the number is limited compared to the affinity-based method. So we have around 304 proteins available for the analysis, and also we have 1,200 tryptic digested peptides, which in the mass spec we mainly measure the peptides and using algorithm to calculate the related protein. So we also did the original peptide level GWAS to identify some additional signals. So overall, we identified a total of around 200 genetic variants protein association, and among which 80% cis acting PQTL and 20% trans acting PQTL. Among these 195 PQTL, so we are interested in whether they can be replicated, of course. So in our independent replication, we could replicate around 85% of these PQTL signals at FDR 0.05. So we then downloaded some statistics from European population to see whether our identified PQTL could be replicated there to have a trans-ethnic comparison. Very interesting, there's 111 PQTL signal which also available in some statistics. We found that 62% of the signal could be replicated in the European data set. And of course, we also did some co-localization of our protein with human disease. And we kind of highlighted 60 gene protein phenotype association after multiple test correction. So around 45 this association have not been prioritized before in the European population, because although in the mass spec-based proteomics, we have a very limited number of protein compared to these affinity-based method, but still we found that among these 300 protein we measured, around 90 proteins have not been investigated before in the, well, is not in the list of the SomaScan or Olympus protein panel. So it still has some uniqueness. So another thing I want to highlight is we did some cross-estimate comparison for this diabetes etiology, because as we know, in the Asian population, usually we are more susceptibility to type 2 diabetes given a certain BMI or obesity levels. So we are interested to see whether the genetic predict obesity or BMI associated with protein with the same pattern in the Asian or in the European. So we found that, so we picked around 41 common protein, both available in our panel and in the Olink or SomaScan panel in the European population. And we found it's very interesting result that the genetic predict obesity could increase 28 proteins among these 41 protein lists and decrease 2 in the European population, but none of them are significant in the East Asia, but opposite direction that we found the genetic predicted obesity or BMI decrease 30 two protein in the East Asian population. So the sample size of these two populations is different. So we also did some sensitivity analysis, took European population with similar sample size as ours, and the results is very similar. So we further did some mediation analysis, which suggests that several specific protein may suppress the effect of BMI on type 2 diabetes, which means the protein may be protective in the media pathway in the European population, but certainly not in the East Asian. So we also create a web server, help readers or those or whoever interested to visualize those genetic and protein or peptide association in the Westlake website. You can look at if you are interested. So as I said, the previous first story is a pilot, and the genetic study is particular focus on genetics. So we are also introduced in the proteomics dynamic change over time, which is the advantage of our study design because we collected blood proteome over the 9 years follow-up at 3 time points. So we are interested in whether this proteome could help predict in Claudia's talk. She interested in the overall, predict human health, and then I think Tianan's group, Xue Cai, also lead another project to investigate the association of the blood proteome dynamics with the metabolic syndrome over time. So in our group, so Jun Tang, who is a postdoc in my group, he is interested in whether those blood proteome dynamics could predict health aging and what's the relationship of the proteome with human age. So actually, we developed a very nice blood proteome trajectory over the 9 years follow-up and cleaned, so and identify those age-related blood proteome. And we found that this age-related proteome are called in the proteome aging clock. This is a name developed by Jun in his manuscript, which still will be submitted soon. So Jun also suggests that this aging-related protein could predict the healthy status, or we can say health aging, because we have major chronic disease, a lot of disease, and also have disease-free subjects. And we found that about the health aging score, fish intake and physical activity could really help modulate this healthy aging protein on this score, which show that this kind of diet or physical activity, maybe intervention, could help attenuate the protein-based aging biomarkers. But there's more exciting work undergoing between the TNS group and our collaborations. For example, because we have collected a lot of stool samples, so TNS group developed a specific pipeline to measure the stool proteomics, or we can call the metaproteomics, around over 2,000 people in our cohort, and connect the blood proteome and the stool proteome together with the gut microbiome. And basically, I think the proteome data have a very kind of represent the functional output of the gut microbiome and show how it links to the human health. So this work is currently under review in journals. So overall, in my group, so we are particularly interested in whether diet and nutrition and how diet and nutrition interact with gut microbiome and the proteome and blood metabolomics and the interaction, how the interaction affects human metabolic health. So in the next step, after this work, we are going to link our diet, these multi-omics, to interpret how to best intervene prevent the human metabolic disease through diet and nutrition, take consideration of this multi-omics data set. So, so it's a very brief talk to echo Claudia's work in the European population. So our work won't be possible without the support from Tianan's very big proteomics lab. So Jian also help a lot with our genetic analysis because we are using the tool he developed like GCTA or GSMR, and also Professor Yi Ming provide founded a cohort study, Guangzhou nutrition health study. So also we have a helpful, quite a few helpful discussion with Claudia and also my whole group. Thanks.
Thank you, Dr. Zheng Jisheng, for your wonderful talk. We definitely hope prevention through nutrition and diet can help us with the disease prevention.
And when I was in Zurich, our team at laboratory of Rudy Episode, we developed a SWAS technology. The major shortcoming is they identify relatively small number of proteins with a very short gradient. I know markers using 1.5 minutes gradient, so that he can run 100 samples every day or even several hundreds. But also there are other mass spec-based proteomic technologies which can measure more proteins. If we first deplete some of the high abundance proteins, coupled with fractionation, we can also analyze up to 1,000, even 2,000 or 3,000 proteins, but the cost is higher. So I noticed you have a paper published in NC, or your paper mentioned you compared 3 methods by mass-based scanning SWAS and SomaScan and Olink. Can you tell me more about this comparison? So have you, I see a Venn diagram, some proteins identified by 2 or 3 methods. Have you quantitatively compared them? And what is the conclusion?
So actually, so I can maybe start, and Mike, feel free to add to this. So the Venn diagram shows, I think it's 185 that overlap between SomaLogic and mass spec. And so we've not done, the paper that we've published is a systematic comparison between just 2, so Olink and SomaLogic. And there we've done a variety of things. So from the observational correlations to the correlation of genetic findings, and then ultimately to the comparison of the disease associations we see for the PKTLs or cis-PKTLs we identify for each platform. So we've not really done this systematically for the mass spec identified proteins. We've done some early analyses. Part of the reason is also because this was a pilot study to look at the feasibility to scale it up for everybody in Finland. So it makes sense to do that once we have it. But I don't know, do you want to add anything to this, Mike?
No, I mean, this is possibly the most outstanding bit to see what can mass spec bring to the table, what hasn't been seen with these affinity reagents. But I mean, the general problem is if we do the 3-way comparison, we are down to 20-something proteins. So what is covered by Olink, what is covered by SomaLogic, what is covered on the mass spec assay. So it's really hard. And the other bit is, and this possibly brings me directly to the last talk we heard, is, I mean, mass spec is measuring peptides, and it's one lesson we have learned is actually those affinity reagents, they supposed to measure a protein, but actually what they do is they measure some isoforms of a protein, and you don't know really what they are measuring. So I think if we can get data together which gets to the peptide level, and then basically look at different isoforms for the same protein header, so to say, and this would be most exciting, I think.
Exactly. Actually, each SomaScan, the antibody-based method only measure one epitope. A protein has many epitopes. So I saw you use a scanning source only measure 500 samples, not many thousands like SomaScan. So probably this is another reason you cannot systematically compare it. But here with Jusen, we have acquired a source data for over actually 20,000 samples. We just submit a paper. Now the technology can go even deeper, means measure more proteins and faster measure more samples. So I think in future we can do more systematic comparison. And for the scanning source, we only need 1 microliter plasma or serum to complete the analysis. So it's really consume very little amount of samples.
Yeah, and as you say, sorry if I didn't make that clear, that's what I meant, is obviously while we're waiting for the rest of the data, it makes sense to do it once that's there. But I'd be interested to hear, so when you say now you can go deeper, so how many proteins have you identified in the kind of 20,000 consistently?
For the 20,000 samples, we still identify 300s. But I mean, if we want, we can measure 2,000 proteins per sample, but with a higher cost. It's not by source; it's by a different methods. We call it TMT. So those for those samples, we first use the antibody affinity kit to reduce the high abundance protein concentration, and then label a lot of samples, 16 samples, combine them into one sample, and do fractionation into multiple samples. And then we analyze each of them, combine the data. So this is normally, it's called label-based DDA analysis. And this can go deeper, but the cost is higher. But currently we are working with another lung cancer cohort in which we really want to identify relatively low abundance proteins for biomarker for early diagnosis, so that we invest more resource to do that.
Yeah. Although Tianan said it's relatively cheap and for this 300 protein method, but still we kind of spend 2 years for the measurement of those proteins in this cohort, right? It's really a big effort between our two groups to measure this stuff. But I think for the affinity-based like SomaScan, they very quickly measure those kind of protein, right?
Yeah, which is, I've been saying we're measuring the 10,000 for a while. So, it sounds good, but of course it is it's a huge undertaking to do that. And you're quite right. Both of the commercial technologies are attractive to people because they're so fast. The turnaround is incredible. And yeah, I had one other question, if I may. If maybe I saw that incorrectly, and I don't know anything really about the stool proteome, but it seemed to me that you identify many more proteins in stool compared to plasma.
Sure. In stool, there are over 80,000 proteins, about 2,000 from human. Because even we wash away the stool, we still find some human protein attached to the bacteria or inside the bacteria. And most of the 80,000 proteins, they are different bacteria. There are hundreds of them. We know the identity, and there are many others. We have at some certain level of genetic annotation, and we realized that those meta-proteomic data, they are much more informative than the counterpart of metagenomic data. Because for many of the samples, Jusen also measure the metagenome, and we can get the functions of these proteins because they are very directly linked to, and they also have better association with the clinical phenotypes.
Yeah,
is-
majority of work in my group are focused on gut microbiome and nutrition. So the protein work is really a piece of work that working together with Ken, which we haven't have any or many publications yet, but I think those data are emerging.
And do you do, sorry, the last slide or when you presented that is a very interesting element, was quite fast, but I'd be interested. So did you do this in just a large set that's part of a cohort, or did you also consider kind of case control comparisons for that? Because it is relatively new, right? Yeah.
Yeah, it's a relatively new technology. And for those that Jason showed, we are part of the Guangzhou Nutrition and Health cohorts. There are about over 2,000 or 3,000 individuals monitored.
I think we have collected 2,000 stool samples from these participants. We actually measure all of them, all the samples that we collect. So including the repeated measure one.
Mm-hmm.
And in another ongoing project, we are collaborating with colorectal cancer surgeries, and we are trying to have a case and control to find biomarkers for colorectal cancers.
Mm-hmm. Oh, really cool.
Yeah, so Claudia, I have several questions for you, or for Mike. So in our PQT analysis, we just our protein list is limited, but still we identify some protein. The association with disease is really opposite compared to the Olink scan, and we found that I don't remember which protein, but it seems that from the biological evidence, maybe Fengzhe is not in the panelist. She's leading that paper, which we correlate, and it's totally opposite in terms of this protein and phenotype association. And from biology, we know that the mass spec basis, right? So I think maybe in future, by link those two methods, if in a simple population, we can calculate some error rate of whatever Olink or some scan. I don't know what's the proportion of error rate, how many protein are reliable or not. Is there any data on that?
Well, my first intuition is to be careful to call it an error rate, because until you've done a systematic comparison and you really know what the target is, what either Olink or SomaLogic capture, you don't really know what specific, biological isoform you may look at. And it could give you some insight to actually compare and find out what we're measuring where we're unsure for SomaLogic. So, I had through your talk, I had this thought of where, our mass spec data is not really yet of the size where it's very informative. However, where proteins overlap, I don't know, you must, maybe you've done it, but the systematic comparison, I think in the GWAS paper that I know, you have done the comparison of the signals for different technologies, right? Because when you look at what's published, that's mainly not MS. So for where you have the same target and the same cis-pQTL, then have you checked? You should see the same disease associations, obviously. Where you have divergence. Is that what you're saying? You see opposite effects? Because it's something opposite that sounded to me same disease, different direction of association, but maybe that's not what you meant.
Yeah, that's what I mean. At the further, correct me if
I think it would be useful if we pull out a few examples. And actually, sorry, I'm not sure whether this is a smaller internal chat or a larger audience, because what we chat here may not be of interest to a larger audience. So forgive me.
We can discuss later.
It's a larger audience.
Yeah.
Okay.
Now that I think that over 500 people are there.
Okay. Yeah. Then we better reserve this.
No, but we can make one point on this, because we have seen this actually as well when we compared Olink and SomaLogic, that we had examples where we see the same genetic variant associated with the Olink measure and with the SomaScan measure, but completely opposite effect directions. And we highlighted one of this in our Nature Comms paper for Alzheimer's disease, and luckily there have been people digging really into this locus already and what we could trace it back is basically what I mentioned earlier. So they measured different isoforms, and depending on the genetic variant, which was a missense variant, you get one isoform which is then soluble, which one assay measures, but the other not. So you get very different information about in which direction the trouble goes, what type of effect might be related to the disease. And the other thing to keep in mind is also more technical. So where you start, if you for instance use any kind of affinity filtering in your mass spec, so you filter down albumin or whatever, you might get some kind of bias in terms of if your protein composition overall changes in the protein, you're more prone to possibly see other things you don't really expect to see. I don't know whether this might play a role here, but I can imagine that the steps before you actually start your protein analysis might also matter in how you interpret the proteins you're looking at. So maybe some of the proteins you measure if you do affinity filtering before might more be of a ratio compared to abundant things like albumin and so on. But to be honest, I don't know how this relates to what we see in olig and SomaLogic. So also how many of those proteins are bound to other proteins like albumin and so on. And so there's a lot of things where other fields like the hormone field with testosterone and thyroid hormones, they have all these assays which distinguish what is protein bound, what is not bound. And I think there's currently these broad capture technologies, they don't differentiate necessarily between the two. You don't really know what you're actually looking at. So this could also all contribute to these discrepancies across technologies.
Yeah, I agree. And I think this is why I think that unless you systematically compare where it may converge or diverge, then we don't really know what good examples would be to kind of drill into, because it's good to have a starting point, right? Yeah. So I think that would be worthwhile. The other thing that might be useful, kind of be good to get Julia's view on this, is the nobody has really, well, we're looking at predictions, one of our interests, or ultimately later prognosis. Now, studies aren't huge yet to do this at scale. However, one of the things that obviously is important in terms of what may predict disease and whether that's transferable across populations, whether that's ethnically diverse or just kind of in terms of the living circumstances, is to have a more systematic description of what influences the levels of proteins in these different settings. And you can integrate the genetics, but you can also look at a whole range of other risk factors. And because in some ways, if it's vastly different, you don't necessarily expect the same proteins to be predictive in different settings. So it would be by definition be somewhat context specific, which is obviously a downside if you're after more generic predictors. So I think that could be a nice joint piece of work to put together the main determinants of proteins where we're overlapping in our assessment, and just kind of compare that if that's of interest.
Yeah, that's definitely interesting. So before we do that, may I ask the technical reproducibility of the measurement by SomaScan? The blood can be stored for different time, let's say some 1 year, some 10 years. Do they have a different, have you checked the stability of this assay?
No, we haven't, and we've just used so this is a historical cohort, and the samples were stored in liquid nitrogen since baseline, so over 25-30 years ago. So none of what we have done, or I say we, the people in Finland who this is an EPIC Norfolk. Actually, in Finland, it is more recent, as I said, and the protocol is slightly tighter, or quite a bit tighter than what it was in EPIC Norfolk. But nevertheless, it's still nothing adheres to the protocol that SomaLogic has in terms of what they would wish for, in terms of processing rapidly and so on. So even in Finland, which is tight, the SOP is tight, it's not processed within, I think it's within an hour for SomaLogic. So we didn't, and we had no other sample to compare to in order to assess that. What we do know is that the influence of technical factors on the proteome is pretty huge, even in Finland, as I said, which was more controlled. I'm somewhat surprised that we see consistency of signals that we identify with these, even with these kind of convenient samples in EPIC Norfolk. And it may be a fact that if you are powered enough, what you see is the robust end. I mean, it's not just the storage or the influence of sample processing and so on, but of course it's also what the time of the day you take the sample. It's people fasted ad libitum, so it's a whole range of, you know, metabolic states that you capture. It is almost surprising that given this, you do see something, and I think it's a fact that what we see is the what do you say, low-hanging fruit. And if you standardize this much better, we would also with smaller numbers see more yield, I guess.
Yeah. So in
But
the
we haven't got the samples to do this, is the short answer.
Yeah. So in the field of mass spec-based proteomics, there was a famous experiment. So someone put the blood in room temperature for 7 days, and then measured by ELISA and then measured by mass spec. And they found if you measure by ELISA, a lot of protein they changed over a week of room temperature damage. But if you measure by mass spec, they are pretty stable, because mass spec we reduce protein to a line and then cut into a small peptides. So most of peptides inside the protein 3D structure, they are pretty stable. So we also, when we analyze the tissues, we also have done such type of standardization, quality control analysis. So since you have a SomaScan and Olink, probably you can do something like this: destroy the blood by putting a room temperature for, let's say, a week. And if you want, we can actually measure those protein by mass specs at different time, and we can do a very deep proteomic analysis. So normally I would expect the higher balance proteins will be relatively more stable, but for lower balance ones, probably they are more prone to be digested or modified, and probably more technical issues. So I noticed you in your Nature Medicine paper, you developed some biomarkers, 70 protein or 3 protein, together with other clinical phenotype to predict disease. So those based on the some antibody-based assay, SomaScan, whatever. So I think if you really want to translate this into clinic, then we need to have a very rigorous QC, different technical issues before we can compare those in different countries, the data from different countries.
Yeah, I totally agree. I mean, Julia, do you want to answer this? But yeah, absolutely.
Yeah, absolutely. I can take it. So definitely we agree. I think one of the things as well that is important to consider in sample handling is not only the stability of the proteins themselves, but the fact that leaving the samples outside can also cause a shift in the composition of what you are measuring itself. And maybe it's more related to things that are being released by platelets and as an artifact of the sample handling rather than something that might be related to the state of the individual when they took the blood sample. And in terms of the project for the isolated IgT, one of the things that we've seen, as Claudia mentioned, it's surprising that we pick up, in spite of all of the technical artifacts and the variability that can be in this measures, it's surprising. So what we've seen specifically is that what comes on top from these machine learning algorithms would pick out essentially what is most informative seems to replicate very well across different platforms. So for instance, the 3 protein signature that we pick up for the specific identification of isolated IgT, we have replicated both in an external cohort using the same technology, so SomaScan, but we have also seen very good correlation with these 3 proteins measured by Olink in a different study. So this really gives us kind of confidence on some of the things that are being very robustly picked up by these algorithmic methods.
That's very cool.
Jenna, I think we should collect some questions for the audience. I don't know where is those questions, but Xiaobing is here. So any other question now from Yajie or Fengzhe or Caixue?
Oh, I have a question. I think I always have a question about how to variate the results of the large cohort. We use the different methods or variate it in a new cohort. I think it's a difficult question. 有声音吗？ Can you hear my voice? No?
Yes, we can hear.
Okay. So, and my question is how to evaluate the results from the large cohort. Just use the different methods or validate it in a new cohort. Okay.
I mean, different purposes of validation, right? The technical
Yeah.
validation, generalizability, depends what your purpose is. If I understood the question correctly, I think
Yeah.
if you, yeah.
Yeah, I think from a large cohort wise, the most important thing is independent replication, different cohorts.
But sometimes it's difficult to validate it in a new large cohort, I think. Yeah.
I think one of the points about you can say validation, people in genetic terms will call that replication. And one of the challenges will be that where we have the largest possible set in a single study like UK Biobank, it would be very hard to replicate outside. So I mean, the availability of other data sets outside, Europe or European centric analyses is becomes very important, particularly at the similar scale, and using a variety of methods. So I think that's one important point. I guess it's partly why Mina is pulling together the existing evidence. However, that's still small in comparison to what UK Biobank is doing currently.
Okay, thank you.
So I have one question here. So I think from Finland proteomics data, we got many interesting biological insights. So will you do some follow-up to validate these findings from proteomics analysis? And the other question is, can you hear me? Yeah.
Yes.
Yes. The other question is, as the UK Biobank, I think Pharma Proteomics project just released the result, the results by do a GWAS and X-Y analysis for proteomics data. So in the future, we will do some meta-analysis between Finland and UK Biobank and like decode, like combine them together. Or they come from different platforms. It's maybe harder to do a meta-analysis to get stronger signal. Yeah, that's my question.
Yeah, I can answer, but I don't want to put anyone on the spot. Mina, do you want to answer this maybe? I don't know whether you're there, otherwise happy to chip in, whatever works.
I'm here, but my connection was cut for a bit. So Yaji, could you repeat?
Yeah. So my question is, so actually we got proteomics data from SubLogic for Finland and the decode, and the UK Biobank use Olink to get their proteomics data. So in the future, will you do a meta-analysis to increase the sample size?
No, of course. As Claudia mentioned, we're currently leading the Scallop Olink meta-analysis to have the largest independent replication dataset for UK Biobank. And the idea is to have large meta-analysis within each platform, first to assess sort of the comparability of the summary statistics across within each platform for different protein targets. And after doing that, we are hoping to meta-analyze both of the platforms, combining the different summary statistics to have an overall picture of what is consistent and what is different in terms of the heterogeneity. And in cases where we see differences, what is the major source of heterogeneity and how that sort of translates into the clinical follow-ups. I hope that was...
Fonda, do you have any question? I just saw you.
Yeah, I noticed that the huge influence of the methodology on the on the proteomics data, but it is interesting that in our article we also compared the genetic background between the different acidics. We noticed that even we have different genetic background and we measured use different technology, and however, the effect size between the identified pQTLs across different acidics shows a moderate correlation. So I think sometimes the identified the pQTLs may be conserved between the two acidics. Yeah.
I have to go in a minute, but I really like your comment because I think people often focus on the differences. And actually, there are quite a lot of similarities, which are good to point out.
Yeah. So I think please.
Yeah, thanks for all your participation and thanks for the very insightful talk by Claudia. Very impressive work. And so many nice cohorts being analyzed at the proteome level. I also understand that you have also quite other omics data, metabolomics and genomics, transcriptomics, which you have no time to talk about today. And also thanks for Jusen for mentioning small portion of his research at Westlake University. And we also have something exciting to share with you maybe next time. And since now already 1.5 hours, Claudia needs to go. I think we have to conclude here. Thanks, Dr. Zhao, and thanks, Mike, Julia, Mai, and Fengze, Xuetai. And then let's meet together next time. Maybe in a smaller group, we will have a more concrete discussion. Thank you.
Thanks so much. Thanks,
Thank you.
everyone.
Thank you.
Bye-bye.
Thank you.
Thanks for all the audience. Bye-bye.
Bye.